To the student. So,
Sure.
yeah, then you can start.
Yeah, please go ahead. Whenever you want me to start, I can just go.
Thank you, sir. So, students, very good afternoon. And we have with us Dr. Parthasarath, who is he is working as a principal scientist in CSIR IITR, Indian Institute of Toxicological Research, in a broad area of computational toxicology utilizing biochemical informatics, computational biology methods, and leading computational toxicology facilities at Center for Innovation and Translational Research. Prior to joining CSIR, he worked as a scientist with highly reputed institutions like the Lawrence Berkeley National Labs, Los Almanzo National Laboratory, and Sandia National Laboratories, USA. He also served as a lead computational chemist and discovery team developing applied toxicologies and product process development at the Joint Bioenergy Institute, USA. Dr. Partha continued his PhD studies at CSIR CLRI and received his doctorate in the computational biochemistry. He has more than 17 years of research experience in applied computational chemistry and biology area, and published more than 100 research articles in Journal of High Repute. And for international patents filed from his research contribution. He has received several awards, including a prestigious Director's Fellowship uh, for Los Alamos National Laboratory in 2009 and the Young Associate of Indian Academy of Sciences in 2008. His expertise includes computational predictive toxicity development, advanced biofuels, renewable chemicals, bionic liquids, and nanotoxicity. So, with very brief introduction, I am welcoming you, sir. On behalf of Srishti, very warm welcome and really honored to have you. Pardon me, whatever information I got, I just introduce you for the students. But over to you, sir. You can start.
Yeah, thank you so much, Magna. Thank you so much. And first of all, first of all, congratulations to everyone for this attending this pretty prestigious biotech innovation, the innovation camp. And this is one of the greatest thing and very exposure at the early stage towards innovation and development, entrepreneurship, and everything so on towards societal applications. So I first of all, I'd like to thank the organizers and Srishti and our beloved Gupta, Anil Gupta ji for all the support and giving this opportunity to share our kind of excitement of doing this research with one and all. So so this topic, when I was yesterday, I got the kind of a list of everyone. I found like very interesting. Like people are mostly from biotechnology, biochemistry, microbiology. So in the talk, initially I made it only for mainly toxicology, non-testing approaches or like computational approaches, how that can be utilized for replacing toxicological experiments, whatever we used for in vivo or in vitro methods, particularly animal experiments. Then I thought of including some more like kind of exciting biotechnology application to you, how one can use this computational approaches. Like right now, this emerging, like you probably hear every day about data science, big data, or machine learning, artificial intelligence. And beyond that, like the fundamental chemistry and biology, how that can advanced in this term and can be utilized for your whatever you do in your research component. Actually, this will give you a preliminary or priori understanding and minimize your time and efforts to achieve your targeted research. That's the kind of a huge advantage of one should use these computational approaches. I think probably my previous speaker should covered about how these data science or computational approaches are utilized in this field. And so I thought of covering a little more aspects on pharmacology and toxicology and biotechnology applications. So with that, like this, as I said, this outline of this talk, like I'll give you very, very brief about our fundamental concepts like basic computational chemistry and biology or cheminformatics approaches used in biotechnological application or biochemistry applications. Second, I'd like to highlight our kind of ongoing work in our computational toxicology facility at CSIR IITR and how we are developing an alternate platform for toxicological testing using computational approaches. And later part, like how the predictive modeling and forced gust interaction as well as some bioenergy application I'm going to cover. And finally, I'll end up with our recent work on computational toxicology about the COVID-19. I think some of you also listed mentioned about drug designing for COVID-19 or base management or many very, very exciting topics. I really very much impressed upon all of your research interest. That's why I would like to touch upon few things which is probably give you some kind of a sense of how one can utilize it. So before I start, like CSIR IITR is the only dedicated toxicology institute in the country, very few in the world. So we are working like kind of a niche area of toxicology, particularly in a very, very broad area. Like we have a system toxicology, health risk assessment, food drug chemical toxicology, regulatory toxicology, environmental toxicology, nanomaterial toxicology. The new addition is our computational predictive toxicology. So all these toxicological domains end up into relevant to translation. It's directly relevant to translation to health and environmental safety, or research for policy, and also we work for industries and providing our services for product safety and testing, as well as designing and safe product and portfolios for industries. So industries, startups, and all those things currently we are developing for, particularly I'm part of a BioNest at CSIR IITR. We are working on various disinfection solution or developing some novel methods for toxicological testing that can expedite the product development. These are the things like we are currently covering up, including pharma products. Okay. So this is our overall background. So computational toxicology is the recent addition in this in the Institute of in IITR, and we have a very three pillars. Like what we want to do in computational toxicology is developing an innovative predictive approaches to address toxicological issues. This is our like kind of a background, like that's kind of a baseline for us. Using that baseline, we fundamentally develop some basic understanding how these biological components and these toxicological compounds are interacting with each other, how they modify the pathways using multi-resolution approaches. I'll touch upon what is multi-resolution approaches in a minute. And cheminformatics approaches are like kind of robust methods that also I'll cover in my couple of my slides, and that use a data analysis and dissemination of larger compounds. So how we can develop that informatics platform for predictive toxicity and developing toxicological databases. And also we provide services to various companies and startups and industries. So this is our kind of portfolio for computational toxicology. So in that, what makes this computational toxicology to be acceptable? Like whatever you do, data or data analysis, measuring like some kind of a compounds, their values, and you see the trend and all those things based on your logics. So the logics behind these computational approaches are really, really strong science behind it. Like probably you may hear about physical chemistry and quantum chemistry. And these are some mathematical modeling, graph theory. These are the concepts are involved in making this computational methodology to understand various events or biological or chemical issues at different resolution. for example, here in the left side lower corner, I mentioned like Hartree-Fock and DFT methods, or some of the methods, we call them as a quantum chemistry methods. That quantum chemistry methods is based on Schrodinger equation. And that Schrodinger equation will solve each molecule or compound that has an atom in it. The atom has a inner components like proton, electron, and nucleus. So we make this equation and solve for each atoms with more accurate level. So that will give you a representation of the molecule. Once you have the molecule, we compute the properties. That properties will compare with the experimental or spectroscopy or NMR or anything. We made them like kind of how they are matching with each other. So then we make these methods are more accurate enough, and that we use for our application purpose, like R&D purpose. So for example, this quantum chemistry has a lot of advantage. One can study the molecules that's more accurate in terms of bond formation, bond breaking, how one molecule can represent in a chemical reactivity properties or functional properties, catalysis, or how they are doing. All those things one can use these quantum chemistry methods. But disadvantage is you cannot use beyond such atoms, like kind of if you have more than like 1,000 atoms, it will be difficult to handle in quantum chemistry methods. For that, we started using molecular mechanics methods like molecular dynamic simulation. That's solving Newton's law of motion, like you know that F is equal to ma. That represents a molecule connected with another represent an atom connected with another atom with the bond distance, bond angle, diagonal angle. We call them, and rather represent in a force field formulation that will give you an idea about the system. So that system will give you a overall broad picture of the molecule in a function of time. We call them as a molecular dynamic simulation. So that will give you, you can study the bigger, like kind of a big protein, like enzyme, and how the ligand binds with the enzyme, and how these proteins are changing its conformation depends on your time, like kind of disease biology. So you can go for many other applications, but the disadvantage, you cannot break this bond forming and bond framing, and a few other aspects you cannot cover using molecular dynamic simulation, but you can study the system in a higher, much broader level. So there is one more recent development called the hybrid approaches, like QMMM methods, like where the enzyme interacting with your active site, you can treat them in a quantum chemistry level. Rest of them you can treat in a higher molecular mechanics level. So that way you can like kind of compromising the factors in which you want to study your particular targeted properties, like how this enzyme, this particular ligand interact with the active site, what are the bond forming, bond breaking taking place, what is the major influencing factors involved in this stabilization of this complexation, and the catalysis, everything you can study. So overall, this kind of approaches give you very close or realistic to your experiments, what you're trying to do in your lab. So that's where the cartoon says. So, and next, I'd like to touch upon a few things, like where this computational capability are applied. So applied in various fields. Like currently, we working on the few red component I have highlighted, like waste management, cleanup, alternate energy sources, antibiotic resistant food science. Apart from that, you probably know about drug design. That's where we are going to talk about more in my talk. And that will, computational approaches are really expediting drug design. Recently, we have seen this pandemic COVID-19, the drug repurposing is most this majority derived by computational or in silico approaches. Okay, so that's where the like kind of you can take the molecules, understand the property, how they interact with their particular COVID-19, the coronavirus protein elements. So all those things people started studying. So that's where all this like molecular medicine, personalized medicine, preventive medicine, gene therapy, drug development. All these are covered under drug development and drug discovery. And microbial genome application, waste management cleanup, climate studies are relevant to environmental sciences. Like if one want to develop an effluent degrading enzyme, like the enzyme or any microorganism, they will go and get that effluent, the industrial site area, and see the microbes which are doing this function, and take that and sequence them, understand how they are go and interact with the particular pollutant. Then you started developing your own way, this expedite this process in a much better way. So that's where currently in our IITR, we are working on bioremediation processes. So we develop these integrated computational as well as these experimental approaches to degrade the pollutants coming from the industrial waste. So alternate energy resources, and I'll talk about it in my slides. And there are very many other aspects like forensic analysis, bioweapon creation, evolutionary studies, crop management, insect resistance. Like you can, once you identify the genes of particular topic and how the genes can be incorporated and how the genes can be cause allergenicity. So all those issues you can deal with the computational or bioinformatics approaches, and how to improve nutritional quality. So what nutritional quality you want to incorporate, and that you can put it in your metabolic pathway level, and that will create create one such new product for improving your food and nutrition of your compounds what you're looking for. And drought resistant varieties, like what are the plants are surviving in the drought resistant area, how to incorporate that gene into the normal like plant. I think somebody also interested in plant engineering. So these where already it's proven, many of the computational approaches are utilized in a very interesting way, an exciting way, or innovating way to provide solution to these areas. So food science, currently we also develop various risk assessment framework for like food safety. If you add additives, so additives are generally considered as a safe element like grass compounds, but many of them, they don't have any toxicological value in it. So what we thought, like we develop an database that will provide you online digital food safety tool and risk assessment for food additives. Many such activities are currently we are working on in the area of food science. So coming back to the topic on towards developing replacing this experimental animal models. So why we need that? Like what is the current gap and challenges in this domain of computational toxicology or toxicological research? So if you see, like this is like kind of a little old slide. This is the data is available so far. There are more than 60 million chemical compounds we known so far that listed in our CAS database, like that's used in pharmaceutical, agrochemical, household, and various personal care products. When we looked at the National Toxicological Program of United States, there are more than 80,000 chemicals are registered. In that 80,000 chemicals, only 350 having long-term and 70 having short-term toxicity. So that means like there is like kind of a chronic and acute is toxicity testing is done only for a very handful of compounds. That also require more than 2 to 4 million for each chemical and took more than 3 years to, because currently we are working in Toxicology Institute. We have a GLP, one of the first lab in government to get the GLP certification. And there also we have seen like some of the tests takes more than 6 months, or some of the tests takes more than 1 year to test these products for their safety. So we know this $2.4 million and over 3 years required for a chemical. So if you want to do for all the chemicals, this much amount of billions required and these many years also needed. So to overcome these challenges, computational chemistry offer a much more rapid and robust platform to predict toxicity. The next of our topic of concern is like how we can replace these alternatives. Like animals, generally we started using in traditionally to compare to test our product or chemicals or drugs, everything. Like we add them like various rodents and non like larger animal models. Like so those how accurate they are. So literature says like they are many of them are not having that kind of what kind of a profile what the humans we have. It's not really matching with the various animal models used in the experimentals, like only 5 to 25% accurate. And some of them like in vitro methods are like 80 to 80% accurate if we develop that particular kind of culture what we are looking for. There are a lot of other things associated with our pain, distress, and ethical issues, and time-consuming post-protocol, high cost, and translational rates, as I already talked about. So there are several animals, like several lakhs of animals or millions of animals used in this experimental testing. And recently, the European Union or US FDA, particularly on India, also we are trying to concentrate more on reducing the animals for some kind of a application, like cosmetic applications. Already animals are banned to use. So slowly it's evolving. So definitely we need to develop some new methods, both in vitro as well as in silico. Like in silico is computational approaches to match and develop some much more accurate level of toxicity prediction. That's the kind of a need of the hour. So we started moving to next generation risk assessment tools. So this slides recently I borrowed from this computational toxicology, the reference cited in the bottom. So we have like a couple of different segment of approaches people are started working on next generation risk assessment. Like the one is like identifying the scenario and how the molecular structure of the compound is you are interested in. So collect the existing data, and the existing data give you a knowledge or expert knowledge that will provide a, determining factor where you can make some like kind of a you can decision for the particular compound is toxic or not. How this compound, which part of this compound is making it the toxicity, because based on already available existing data, if you have some particular group is giving some alert, so you see the molecule of your interest also having that particular group, then it will clear cut having a group or read across approaches. That's what we call them. So that will group them into the particular category. So that category immediately they identified as a toxic compounds for various toxicity endpoints. So then we can also go for read across approaches to identify analogs, and what these analogs, how their property are suitable for the application you are interested in. So that's where the second part is coming from. Like if you create a new molecule, what is how they are different from parent, what are the metabolite it going to form, and where it go and bind, what is the pathway it's involved. So it's a kind of a sequential steps we started doing for next generation risk assessment methods. Actually, I'm going to cover this various aspect of this next generation risk assessment in my slides through our couple of our research projects, and how the another approach is like application of ab initio approaches like target testing, and we also calculate the biokinetic refinement like PKPD. You are talking about like pharmacokinetics, toxicokinetics, pharmacodynamics, toxicodynamics. All those in vivo, in vitro data is available, and also we can try to match it in silico methods. And this is kind of overall picture where we actually we search and retrieve the existing data of the particular chemical, and identification of structural alert, and to identify its activity and indicate their toxicological component in it. So then we make that to overcome these approaches. So we make them in a very group and read across methods to develop some new molecules or identify the existing molecule or repurposing the drugs or repurposing the compounds. Then we develop a model which we can predict the activity, toxicity, or like so on and so forth. So based on available data. So when the data is not available, then it's a problem. Like if you similar molecules not available existingly, then it's become kind of a challenging issue. For that also we have methods to overcome because based on your quantitative structure activity model develop, that will give you a confidence of your model to predict your new molecules. So mainly we trying to remove this where this toxicological alerts are coming for the particular toxicological endpoint or mutagen endpoint or carcinogenicity endpoint. Some few examples I'm telling you. So that's where we omit those molecules and go for a safer alternatives. Okay. So this is kind of a next generation risk assessment methods using in silico approaches. So I like kind of sorry, I missed a couple of my chapters we published on this topic. That's where this figure diagrams have been taken, and so advantage I'll highlight very quickly. They are low cost, high throughput process. Like you can study like 100,000s of chemicals within no time, like within a weeks or so. It depends on the accuracy of your model. And also they provide a mechanistic understanding. So mechanistic understanding experimentally, it's kind of sometimes is difficult or like kind of time consuming process. But here, based on the structure, you can understand at the molecular level how the compounds are interact with the biological system. And you can also overcome by this toxicology aspects by safe by design approaches, or you can re-engineer or repurpose the drug, which the particular compound which has this nitrogen group which connected with the particular moiety is making this problem of various toxicological issues. Then we can replace that and we can retest and see whether the molecules are suitable for our target purpose. So that's how we can prioritize the lead, very fast identification of potential hazards or chemicals and drug extra. And finally, we have to integrate with this in silico, in vitro, in vivo, that will provide a better, much more advantageous model for toxicological prediction. So this is the general trend for drug discovery and development. Identified that this is protein isolation takes like almost 2 to 5 years, and another find a drug effectiveness, preclinical testing, formulation, filing. It's almost 8 to 15 years of process. So the new revolutionary technology like gene chip, now organoid chips are also coming up, bioinformatics, visual screening, docking, in vitro, ADME testing. ADME is absorption, distribution, metabolism, and excretion, and toxicological testing. That will make this process into much more faster and sophistication, and maybe within 2 to 5 years, or sometimes lesser than that if it's a known compounds are existing and you can repurpose them within very short time. Like you can develop a model or compounds to expedite this drug discovery processing by replacing this in various in vitro or in vivo animal models. So very quickly, I touch upon the in silico model has a lot of advantage. I told you about metabolic pathways, receptor ligand interaction, and disease models also possible, decrease potential adverse effects, and understanding its physiology and possible to generate virtual patients. Actually, I didn't touch upon, there is various system level approaches also people can currently working on network pharmacology. That's where we can also like understand the various adverse effect and improve upon the drug component. And our disadvantage of the in silico methods are simulation models reflect what is known. So the mechanism is not clear, the model might not suitable for your purpose of your work. So that you need to be very careful about it. And the another one is best use of model is simulation of cell responsiveness, disease induction, factors involving generating chronic diseases, predict drug treatment efficiency, adverse side effects. These are the way you can very carefully choose this problem and integrate with existing in vitro or in vivo methods. definitely this is going to be a advantage method for drug design and development. Not only that, also safe product and process development. So a few example I'm going to show from our research. Like one example is chemical safety. There are, as I said earlier, like more than 80,000 chemicals are currently registered, but we thought of going for like high product volume chemicals. Like 1 million pound or more are produced and imported every year, about 3,000 chemicals. So they are used in many applications, like our day-to-day like consumer products. So those products are relevant to various health issues, like they cause skin allergy. Some of them cause respiratory problems, or some of them are like known neurological dysfunction, like modify blood-brain flow or blood pressure or migraine, headache. Finally, some of them cause like stroke, or some of them cause long-term issues like hemorrhages, and so on and so forth. And some of them are caused like chemicals enter into the body and to various routes, like mode of going into the system, and some of them they can cause blood-brain barrier, and that cause defects in our body and impair the organ and system function. So that's why we are want to study the toxicity of those chemicals. So the systemic toxicity are listed on the one side, and organ-specific toxicity are listed on the other side. There are many toxicity endpoints and many mechanisms. It's a very tough problem, even though like whatever you are doing, experimental animal models or computational in silico approaches, it's very like kind of yeah, broad field, and also need very in-depth or deeper understanding of each one of them to and also they are challenging, and how we can develop these models. Like for example, acute toxicity, we use like experimental animals like zebrafish, or some other alternate animal models used for aquatic toxicity, subchronic toxicity, genetic toxicity, carcinogenicity, developmental toxicity, phototoxicity. So that's like how the skins are behaving, like particular chemicals what we are currently using. So there are n number of things, but depends on your applications. You can pick like few of your acute toxicity models or chronic toxicity models. You can go for your currently for developing in before starting your all complete level of toxicity. You can go for very pick and choose a particular endpoints or desired application. You can select that particular toxicity endpoints. That's where we started doing our research. So one such example is genotoxicity. So as you all biological life science students, you know about genotoxicity and mutagenicity, right? Like potential to interact with genetic material, modify some DNA alteration. And because these chemicals are DNA reactive, they cause mutagenicity and potential to change genetic properties, causing heritable and genetic mutations. Some of the methods conventionally we use for chromosomal aberration, micronuclear formation, or sister chromatide exchange, or comet assay, understand the DNA damage. So these are the like kind of experimental approaches we currently use for. But those experimental approaches are like, as I said earlier, so these are mostly are in vitro methods, not in vivo methods. But still, we want to develop a model, and you can also, many of them all probably started using animal models, also carcinogenicity or like mutagenicity models. So we want to develop a predictive model for genotoxicity. So why? Because Reach also suggested to use in silico approaches. So we started using QSAR. QSAR is like quantitative structure activity relationship. That means like if you know a structure, that already know to act upon a function or property. If you know a structure, that will have some function, right? If you know a similar structure, that will also pretty much behave the similar function or similar property in which the parent molecule behave. This is the very thumb rule, like similar molecule behave similarly. That's the structure activity relationship. So if you know the structure of the molecule, so you predict various properties and the property relevant to some toxicological aspects. In QSAR approaches like quantitative structure activity relationship, you take a series of chemicals and known toxicological endpoints like IC50, inhibitor concentration value, or EC50, effective concentration value. So this already known experimental value, it's available, take them. And you take this computational approaches, taking these compounds and predict some properties. Like, for example, how they ionize these compounds. What is the ionization potential or electron affinity or some so on and so forth. So we can calculate so many properties of your compound. That compound will match with the existing experimental data. That will give you a relationship between how these properties relevant to the experimental toxicological values. So once you have that, you will create a model. So we divide into two categories, like test set and training set. So what are the known compounds? They will make a model using test set. Then we train them with the already training sets of those compounds, whatever we have taken, and we test them with what is not known compounds, or sometimes we known compounds also, we test them how it's matching with your predictive models. Once this predictive model is ready, then we can go for a prediction of the new compound with more confidence of your developed model. There are several QSAR tools currently it's available, and because right now the pandemic, like people started using many of the computational aspects. So you can at home, you can test, some of them are commercially available, some of them are open sources, many of them are open sources like listed in this here. So you can use these QSAR models, and you can also start working on in silico approaches to predicting the properties or toxicities. As I said, structural alert, right? Like, so how this robust model, like if you go for one-to-one product, like understanding one compound in much deeper way, that's time consuming, but that is the best way to go about it. But currently we have thousands of molecules, like 10,000, 20,000 molecules within shorter time we need to screen. So what we did is a method called structural alerts. So in the genotoxic properties are relevant to particular structures. So if the database containing this kind of a genotoxic compound with the known of this particular structure of various AIM test or some other machine like mutagenicity prediction, that's where they tested and they created as a database. If you put your molecule of interest or compound of interest into that particular database, that will search the entire database of your interested compounds and pick the molecule which has a structure in which it's causing the particular genotoxicity or mutagen, immediately give you an alert. Oh, you have this compound. If this compound has this particular moiety, that's going to create a problem to you for genotoxicity or mutagenicity. So immediately we'll identify and omit these molecules, and we use other molecules for your drug development or any other product development. So these approaches, we call them as a machine learning models, like the QSAR machine learning models. In that, like, Vega platform, we started using this for carcinogenicity and mutagenicity prediction. There are 4 each models are available. So in these 4 or 5 models, whatever the 8 models or like 9 models I have listed, like mutagenicity 5 and carcinogenicity 4, so they have its own level of accuracy. So if you're, as I said earlier, if the molecule does not having a database, it's going to predict very wrongly. So you need to be very cautious about it. So what we thought, like kind of we going to predict some kind of a known like 130, 140 chemicals or unknown toxicity values, which has another 40 compounds having a known toxicity of genotoxicity or mutagenicity. So in that, we want to test these molecules in this machine learning QSAR tools. We found various different results. So we don't know which one is more accurate and all. So what we thought, we systematically started using developing genotoxicity approaches in which we make a benchmark of this known QSAR models. With the very careful analysis, we pick such model in which it will give you a better accuracy. So this work, like a couple of I think year back, we published in one of the special issue of Theoretical and Computational Chemistry. So we have taken like 131 compounds from Reform database. I'll tell you, like you can also take some various databases of your depends on, because many of you guys are going to develop some various innovative methods or product or process. So first test them whether it's going to have any environmental impact, health impact. So that's the reason why I think it's very thoughtfully they made it in a various composition of this schedule of this program. So you can take that and test using this. Already open resources are many of them available, you can test them. So here what we thought, we want to get that 131 commercially applicable compounds which is used in various consumer products. We have taken them, but when we started predicting, we don't know which one to use. So what we thought, we went back against some of the databases. So those databases, we have picked those compounds, and using this machine learning tools like mutagenicity, mutagenicity, and carcinogenicity, we predict them. Then we did an analysis called a five-fold analysis, like specificity, sensitivity, accuracy, positive predictivity, negative predictivity. In that, which model seems to be much more higher level of prediction, we pick them and we predict the toxicity of this 131 compounds, what we are intended to do. In that, like there are almost 40 compounds having this known values. We match up with them, and our results are highly matching with those known toxicological toxicity data of those compounds. Then we started predicting the risk assessment, which are the molecules are safer to use, which are potentially harmful, which are highly safe, or which are like nominally safe, which are harmful or most harmful. That's where we classify them, and we also refine this model that will give you a best model for mutagenicity and carcinogenicity. This is kind of a very robust approach, like because if you go for individual molecule testing, you need to know a lot of theory behind it, like, but still I strongly suggest you to go and read and develop your knowledge. That's very important. But if we for like kind of a time factor and all those issues, you can go for understanding of how these concepts are working and go to some kind of a robust methods currently available, these QSAR tools, to predict chemicals and their safety. Another one is like example, currently this work is published in Chemical Research and Toxicology. You know all about 3D printing, right? The very exciting field. But when we are using this 3D printing, we use the materials like fused flame materials, right? Like that's heated upon sometime. But when it's heated upon, there are various particulate matters, like volatile organic compounds are getting released. When you're using in a closed environment, that's still it's okay. But sometimes open environment or in later points, some of them are like within very lower temperature also the chemicals are getting released. So we want to make a risk assessment platform using computational approaches by integrated QSAR and adverse outcome pathways, and that predict this toxicity of the fibers. So that way we can omit those fibers and develop some safer fibers for our users. So there are genesis of this project, like there are more than like 700 million worth of industry of 3D printing right now. There are fused filament material, plastics, metals, ceramics, powders, liquids, or even living cells. Currently, we are using it. That's our biological component we are using for various toxicological testing. But when we are using this material printing, you definitely need to do risk assessment and develop some policy framework for their safety. So I'll tell you like this graphs here shown here on your right hand side. We compared these two products, like it's from earlier work published in Atmospheric Environment and Journal of Occupational Environmental Hygiene. So they compared this PLA, polylactic acid, and ABS, acetonitrile butadiene styrene. This is the fibers used for. They see that, like if you see that ABS had higher emission rate, right? And also this one, like ABS also showing like ultra fine particles or higher number of ultra fine particles are getting released. So here we show some of the particles. So what we thought, we want to do a systematic analysis of all the ABS, taking the ABS filaments, and what are the volatile organic compounds or like ultrafine particles are released upon heating. So we collect them from the literature, because since I mentioned in the next generation risk assessment platform, we take them and collect from the literature what are the chemicals are getting released. Then we started predicting their absorption, metabolism, digestion, excretion, and toxicity, and compare with some of the animal model data available from National Toxicological Portal using QSAR methods. Then what we did, like once we characterized this ADME properties, then also we found out what are the adverse outcome where this chemical compound, like particularly ultrafine particle, go on interact. Because I mentioned one time on metabolism, right? Metabolism and pathways. Go and particularly target a protein or target in which it caused some adverse impact or inducing this toxicity. So this is what we the study methods we have chosen. So here for toxicity profiling, this QSAR model we used for inhalation because it's mostly going in respiration and going into the oral toxicity, and long term how this carcinogenicity is going to impact them. And the metabolism wise, like how they are particular endpoints like expert-based system reveals you, like thyroid toxicity, nephrotoxicity, teratogenicity, hepatotoxicity, some of the toxicity points we have selected using our our tools and techniques what it's available with us. So this slide shows like among the selected like 41 compounds, the 49 are highly toxic based on this oral toxicity, and 51 are extremely toxic. And we kind of compared with the experimental data what is available in National Toxicological Portal with various rodent models is available. That's also kind of indicating the similar suggestion for what we predicted for oral toxicity. And there are few discrepancy also we found, and we also found out like male female has a different effect. That's also we covered it up, and that will also be an important point for you to understand and detailedly discuss on your study if you want to pursue this work. And also we predicted various toxicological endpoints what about these chemicals, and they very clearly says these are the, except very few, many of them are creating various toxicological endpoints. ADMET also shows like a similar pattern, like many of the compounds are highly absorbed, they can cross the blood-brain barrier, and some of them are they can related to neurological disorders. So that we find out from our pathway analysis. So this will give you a clear understanding of how these compounds are impacting the human health. And sometimes the environmental health also very relevant, like when it's going into the environmental through various channels like water or air, that's going to create problem to environmental safety of animals or any other relevant area which is going to be impacted by these chemicals, like the industrial pollution. That's the main cause for this 21st century. So then we did a like kind of a pathway analysis. Like we use the Campbell, the Panther. There are a couple of softwares are available. So using that, these are open resource. You can go and clearly test by yourself. That's why I'm covering these topics. So take that particular chemical. We went there and test them, like how they impact and which protein it's targeting. So total of 184 protein targets were identified for these chosen chemicals. Then we did an analysis where and which exactly it's go and binds and which pathway it's impacting. So that's give you, oh my God, like what's going on. So we need to be very careful. This is clearly elucidate, like you need to develop and safer fibers for usage. So they are impacting immunological signaling, neurological signaling, cardiovascular signaling, hematostatic signaling pathway, and oncogenic signaling pathways. So in that way, we covered a systematic study in which we collect a fiber what is used for 3D printing, collect the chemicals, understand the ADMET properties, and the pathway analysis. This way, you can make a risk assessment framework for safer design of new chemical fibers or existing fiber, how they are safe. So that's where we started this study. Okay. So just I'm switching the gear very quickly. In like 10 minutes, I'm trying to finish it off this talk. So the biofuels, like during introduction, they mentioned like I worked in Lawrence Berkeley National Lab and Los Alamos National Lab, where I started working in biofuels. So why this theoretical chemist or computational chemist interested in biofuels? That's where I'm going to touch upon how computational chemistry is expediting the discovery in this particular alternative energy energy area. So biofuels are really need of the hour due to depletion of fossil fuels and increasing the price and all those things, as well as environmental climate change, all these issues. So we not only we need to replace this existing fossil fuel based petroleum refinery, we also completely replace whole barrel of oil. So in that lignocellulosic biomass, particularly from the the bark and trees having the lignocellulosic biomass, that has the capability, it can form many type of gasolines and various products and chemicals using different routes like catalysis, fermentation, and so on and so forth. So you can develop n number of products, which is very closely you can match the replacing the barrel of oil. The challenge is the structure of the lignocellulose. Lignocellulose is the name suggests like the biomass combination of lignin, cellulose, and hemicellulose. Cellulose is the linear chain of glucose conjugated together. This more polymeric structures is really tough structure to break. And apart from that, it's decorated with the lignin and hemicellulose. It's giving less access for us to take the cellulose, convert into glucose further into like biofuels. So for first thing, what we thought, we want to understand this cellulose, how they interact with each other, and what are the stabilizing forces. So the cellulose in nature is 1-beta cellulose, they call them, that's formed like linear chains, conjugate like hydrogen bonded with each other and stacking with on the top of each other. When we use like particular chemicals like ammonia, like Apex treatment, that will change this cellulose 1-beta into cellulose 3. So we want to understand how their stabilization factors are differing. So this is one of the first work we elucidate the role of hydrogen bonding and stacking difference in this cellulose 1-beta and cellulose 3. That will provide like this hydrogen bonding and stacking patterns are changing in the cellulose 3. That is less stable compared to cellulose 1-beta. So if you want to do a new process, you have to destabilize this polymer structure. And also we found out another interesting aspect is lignin. Lignin is polypropylene ether. It's a kind of a very diverse network. Like DNA, you know what are the bases and base pairs, how they interact with each other. But lignin is depends on the the diversity depends on its where this plant and trees are grown upon. So it has different type of composition. Or if you have a sunlight on one side, it's a different type of lignin, and the other side, it's a different type of lignin. That's how it's complicated. So in order to develop an efficient methods, both techno-economic approaches, you need to understand the lignin and remove the lignin very quickly. So we made a study how this propensity mapping, amino acid propensity, right? The amino acid propensity. Similar to that, various combination of these integrating components, how this bond strength varies. So using computational approaches, we developed a profile of what are the bonds in this lignin and how easily one can break this and what are the product you will get also. Because this is one of the interesting product which can replace the aromatics available in the air, like most abundant component to replace this benzene, toluene, and xylene. So if you develop the product from lignin also, it's more value and add value to your biorefinery process. So what we made this kind of a profile, and some of our colleagues like metabolic engineers, what they did, okay, your computation work suggests like the weaker bonds and weaker compounds. Why can't I do some manipulation in the molecular level at the plant, so I can make that only one type of lignin or one type of this bond present in this plant, So I can use like less dense process, make it more efficient way of generating biofuels and advanced chemicals, right? So that's where this work paved a way for this kind of new approaches. So in the where I worked is like in JBA, so Joint Bioenergy Institute, we started converting this plant into advanced fuels and chemicals. So for that, we thought of developing new, I mentioned ammonia fiber extraction methods. We thought of creating using ionic liquids. Ionic liquids are combination of cations and anions. The imidazolium and acetate, the marked here, are the more suitable compound for biomass degradation compared to any other like some of the acidic solvent or basic solvent. And it's also having a lot of applications. So I'll just very quickly tell you, like, this is the process, like taking the biomass and add the ionic liquid, heat and stir for a few hours, add anti-solvent, and you will get that sugar. And it has provide higher accessibility, decreased lignin content. These are the advantage, but the process is ionic liquid is very expensive. 1-ethyl-3-methyl imidazolium acetate is also coming from the petroleum industry and the cost. So we thought of creating a new method or process of using computational approaches. So one of my colleagues currently is professor in Kentucky University. He is a chemical engineer. So he want to know how this process can be engineered much more efficiently. So we thought of making combinations of ionic liquid and water in which the minimal way we can disturb this, so that will reduce the cost of the ionic liquid used for this process. So we carried out a molecular dynamics simulation of various composition and see how these chains are breaking and how this enable this process more efficiently. So then we thought of making not only the same imidazolium acetate, we thought of making a biocompatible ionic liquid that where we can integrate this process in much more efficient way, because this process done in like pretreatment, saccharification, and fermentation. These are three processes, like remove this component like lignin, hemicellulose, and cellulose. Then you put a enzyme to saccharification. Once you get this sugar out of from saccharification, then you go for fermentation. These are three processes. If you make a compatible ionic liquids or compatible solvent, you can do it in one reactor. So we thought of designing a new ionic liquids with the biocompatible ionic liquids, and we provide a fundamental understanding how they interact with this biological component like cellulose and lignin. And this is where I thought about the QSAR, like this kind of general framework of QSAR. I'll just skip this because I already mentioned to it. Like because QSAR is give you kind of robust way of screening many number of chemicals in a very short time period with the desired properties. So these many number of combinations of ionic liquids are available based on the cation and anions, and we want to select some new ionic liquid. It will be difficult. Like, so we thought of developing a new protocol where we can compare with the existing basicity acidity available for various solvents and develop our theoretical basicity and hydrogen bonding acidity. And we developed a property relationship and validated them with the existing solvent. That gives us idea about how the molecule having a particular type of property showing a good solvent for biomass pretreatment or any other way. So this is one of the very interesting work, like kind of a startup it's created from this work. Like lignin, as I told, like it's a complicated structure. Mostly they used as a waste in that thing, but you can generate a lot of value added product. So what we thought is like converting this lignin into ionic liquid and used for biomass processing. So my colleague is organic chemist, Professor Arun Socha. He asked us like, hey, computational methods can be used to design molecules. Can you design some few molecules from lignin that I would like to synthesize them and test their efficiency. So that's where we developed new compounds like during this process, and that is used for developing, pre-treating this biomass and more efficiently. That's created a much more innovative idea of closed loop biorefinery concept, like taking a waste product from the biorefinery like lignin, convert them to ionic liquid and utilize for biomass treatment. And not only for biomass treatment, this ionic liquid we can generate from lignin, also used for many other applications. So I'll skip this. Like another very interesting chemistry we played with here, like reducing the pH of this choline lysate used in higher pH. If you reduce it in using neutral pH, we can use it for a single reactor. And another one is like if you develop an ionic liquid which is used in a room temperature, that reduce almost 80% of steam requirement required for the biomass processing. So we developed a new, this is a couple of days back also, this US patent got accepted. Sorry, this very old slide, this published in biotechnology for biofuels work. And this work, this compound is very less ionic liquid with more water, and in room temperature, particularly 30 to 50 degree, it can convert biomass into biofuel. Okay, so I'll skip this. And coming back to again toxicology. So toxicology, I mentioned all the QSAR studies, right? So now we thought of understanding how this toxin interacting with the biomolecules like the proteins or DNA. So the DNA, it can form like intercalation through major groove, intercalation through minor groove, that one can study if you want to develop a new compound like new molecules, whether that will induce any toxicity or biomolecules. So this is some of you guys are interested in developing diagnostic or sensors, right? So this work I carried out in Las Alamos National Lab, like the tuberculosis, like the tuberculosis has its cell wall has a lipoaminoglycan. So lipoaminoglycan can used as a biomarker for the disease, as well as this is also inducing a pathology, more pathogenic if you has a Manos cap. So what we thought, like we want to develop a biosensor sandwich assay. Recently, it got like R&D 100 award in United States, as well as now we expanded into universal bacteria the bacterial screener, like diagnostic tool for screening this tuberculosis. So the lipoaminoglycan present in the microbacterium cell wall, it's containing Manos cap. So in order to develop a biosensor, they want to know how they will capture this lipoaminoglycan. It's a gigantic structure. What we did is I developed a model of the lipoaminoglycan because the structure is only sequence is available. From the sequence, we developed a model of computational model, a three-dimensional protein PDB model I'm showing here. So in that, we have taken that and we find out how they interact with the membranes. So these membranes can be used for capturing this, and that can be used for your diagnostic development. And there's a kind of multi-resolution approaches we made it, like how interact with various membrane and what are the toxicity also induces, how they penetrate. It's a pathogenesis, like if you have a Manos cap or without Manos cap, how they induce the differences in the pathogenicity also we have studied using multi-resolution approaches. And currently we are developing several databases that provide a platform for alternative testing capability. And very, very quickly we can search the database and understand toxicity of these compounds of various relevant work. And finally, I touch upon the COVID-19, right? Because you've studied all the non-tested models or next generation risk assessment models, like many of the compounds are fails in the preclinical or clinical trial. Almost 90% will fail. So that's where the computational tools are providing understanding where you can develop a model before itself, avoid those like toxicity or adverse effect impacts. So if you know hydroxychloroquine recently used in COVID-19, that also having issue with some kind of heart diseases or kidney diseases, what we thought is developing analogs for hydroxychloroquine with more augmented safety profile. So this is the structure of the COVID-19. It has like protein, structural protein, non-structural protein. There are spike protein, envelope protein, membrane protein, nucleocapsid. I think probably you all know about it. And we selected one particular protein from the membrane, and we selected analogous approach, like hydroxychloroquine, chloroquine having a structure, and we found out from the zinc database what are the possible matching structures from those hydroxychloroquine and chloroquine. We analyze their drug-like clearance properties and the toxicity screening and molecular docking and simulation. Finally, we picked like kind of almost 30 compounds each level. It's got reduced. Finally, we around 6 compounds we got it for replacement of this hydroxychloroquine and chloroquine. People can directly go and do this testing and go for drug development process. Okay, so this recently published. And finally, I'd like to conclude. There is a great advantage using in silico approaches. Like you all agree with that. Like I covered some of the topics. Sorry, I'm kind of rushing to give you a lot of like insights. That's why I'm very sorry about it if I couldn't detail go into some of the aspects. But they are really great advantage using these methods and this toxicity to ensure safety use and minimize the animal and resources used in toxicology testing. And that's we support this 3R: reduce, replace, and refine. That's where we currently working on. And if you integrate these approaches, having huge strength, and that's where currently CSIR IITR we are concentrating to increase reliability and confidence of our prediction and methods. And these computational approaches, as I pointed out some of the area, provide new solution, provide diverse applications, right? Like more reliable prediction across many domains. And finally, we are merging various our toxicological aspect with regulatory framework. So such model will provide a cost and time effective way to predict toxicity as well as create a safer world. So finally, I end up with quoting this Sidney Brenner's quote of Nobel laureate: "All biology is becoming computational, much the same the way it has become molecular. Otherwise, low input, high throughput, and no output science." And I'd like to thank my team and the entire funding agencies and various mentors who supported me. And thank you one and all. Once again, I thank the Honeybee Network, the Srishti, and the organizers and Biomass for this opportunity. And I'll happy to answer questions to you in another 10 minutes or so. Yeah. Thank you so much.
Should I ask the question right now? Like,
yeah.
Yes. So, sir, I have two questions into this. There are. You have mentioned about 3D printing, right? So there have been cases where the 3D printing filaments has been used from the waste plastic. So if we see the scenario right now, the plastic hospital waste or PP kit waste has been increased so much. So do you have any take on this? And second question is computational biology and waste management. How would you like elaborate on this?
Yeah, I told you, right? The waste management, one example I told you about lignin. Lignin is used as a waste. And the same thing, like you rightly said, the reuse of plastics. Similar way, we can design some new microbes or enzyme using computational approaches. Already existing plastics, right? Some of the microplastic and nanoplastic, how we can degrade them or sometimes detect them. Also, we can use this computational approaches. So the using the waste plastics are is again, it's a kind of a interesting reuse method. But
recycling method. So we are recycling the filaments.
Pardon?
We are recycling the waste plastic filaments into 3D printing as a raw material for 3D
Yeah,
printing.
there are many ways people are recycling it and reusing it. But you need to be a little careful about the process. That's it. If you're careful about the process and what it's meant to be used for, if you clear about it, then I don't think it's a problem with you. And also waste management side, there are we have, it's a huge problem currently. Various research groups working on developing effective process for remediating those issues, and computational like metagenomics, as well as like designing some new novel enzymes which can easily degrade them and convert them into value added product. That's where many of our research group, including in silico, in vitro, and various methods, are currently we focused on. That way we can use it. Like example, like currently we are working on metase and petase. We want to understand how they degrade this PET components. So in that, like once it's degrade, what are the product we are getting? That product, how we can recover them? That's where we are focusing upon. Okay.
Okay. Thank you. Thank you so much.
Yeah.
Excuse me, sir.
Yes, Ayush.
Yeah, sir. Right now you mentioned that you are working on metage and petage over there for the plastic degradation of PET, sir. I was also interested on this topic, sir. Can I go with more inside of this one topic, like the work whichever you have been pursued and all?
Yeah, can you repeat the last line? I can't hear you.
Means the work, how much work we have done, like what are the things, information you have regarding this one? Because I was also interested about this metage and petage for the research work, sir.
Yeah, there are literature available. Actually, you can go through some of the literature. Maybe I can share with you. If you write to me, I can share those already existing, very, very particular in this topic. And I can share those details with you.
Yes, sir. I will lie there, sir. How can I get there, sir?
How you can get what?
No, miss, how can you miss? How will you send it to me? Like.
Yeah, you email me. I think you can get Magna ma'am to share my email. I think in the first slide itself I showed you. partha.rom@iitr.res.in. If you email, I can share some of the literature to you. I just typed my email on your chat box. Hello.
Yeah. Thanks, sir.
Thank you. How we can overcome the data limitation when we are working with new virus protein, especially to find antagonist, antiviral peptide against such virus proteins. Okay, it's a kind of, if you want to computational biology, is it necessary to first data from all sources collected? I mean, databases you access first, right? Oh, so many questions are coming. QSAR modeling in some among the programming language is necessary. So I can share some of the QSAR links, also some of the slides to you. And here the interesting question is like vaccine development is mainly focused on antiviral peptides. So yes, data needs to be collected if you want to do some kind of a rapid design of some drug and drug components. Like there are various ways, ligand based approaches or structure based approaches, if you go into drug design platform. Other than that, sometimes like data is really essential, and databases is like, first you have to see what is the prior art is available, prior knowledge is available. Based on that, you can see the gaps and challenges and how you can take up further paths. So that's where it's better to see the literature and data first. Okay. So, but I'm not getting your question the where you're typing, because it's like too many questions are popping up. And we want to do computational biology. Is it necessary that first data from all resources is collected? I mean, a database has been accessed first. Yeah, it's a one way to start this process, or one way the research question is very, very important. Like what you really want, what will be the outcome you really interested in. Based on that, you can plan your work. It's not necessary you need to go a database search for everything. That's one point. And if you have that particular problem which requires that kind of a work study design, definitely you go search the database, understand what are the components it's there, are already known, because you don't need to waste your time in designing something new which already exists. So that's where I started using it. Okay.
Good afternoon, sir.
Yeah. Yes, Aditi.
Sir, I want to ask that for the toxicological research, why don't we direct approach to the QSTR, that is quantitative structure toxicity research? Like why we first go for the QSAR and then QSTR?
No, it is all same, like QSPR, QSTR, QSAR, all going to be the same. If you are using for particular endpoint of toxicity, you are going for QSTR. It's only terminology difference.
So means they are all like equivalent.
They are all same principle it's working on. If you're going for a property for material science, you go for some property prediction and property matching with the using this QSPR approaches. If you QSAR approaches, you go for biological activity or chemical activity or something like that, then we call them as QSAR. Here you connect with various toxicological endpoint and toxicological things, then you started calling them is QSTR. That's it, right? It's all working on
I was
pretty much same principle.
Yeah, I was like QSTR is a part of QSAR or something like that.
That's how you coin them. That's matter for me. It's like all are in us working on similar principle. Where it's field you are applying, we started calling them in a different way. That's it.
Okay, so thank you.
Yeah. Do bioinformatics and computational biology go hand in hand? Actually, computational biology is a broader term which includes bioinformatics as well. Bioinformatics mostly on sequence analysis, comparison, phylogenicity, and all these informatics approaches relevant to that, then we call them as a bioinformatics. It's all terminology is kind of a term, depends on what you're using for. So you don't just distracted by the kind of a terminologies. Understand the concepts. Then concepts give you the clarity of the research what you're going on. So because the various people started calling in different way how they started using it, right? Like so computational biology is a broad area in which bioinformatics also part of it, and bioinformatics also is sometimes is kind of analogous to computational biology. It's a kind of context specifics. So don't worry about all the terminology. Just focus on your concept and the research what you're driving, which is driving you. And I strongly suggest like don't just look for only your expert area. Try to integrate with other disciplines. Like probably you need a good chemistry. Probably you need some kind of a physical chemistry understanding. Probably right now you need a new domain of data science or computational approaches to integrate with your current R&D. That will give you better results. Like it's not only a single way to go and provide solution. Probably you need to integrate. Currently there are complex problems. Complex problem need a very, very systematic understanding and comprehensive way of approach to solve the problem, not a single route to it. Probably right now various interdisciplinary or integrating approaches is going to give you a better results. In silico optomers, what software should I use? What theoretical knowledge should I have to design it? Oh, right now, I don't know exactly tool used for optomers. Probably I can share the details with you if you send me the email. Is there any more questions?
Hello, sir.
Yeah.
Sir, how does nanomaterial toxicity affect on brain, sir?
It's how it's going to cross the blood-brain barrier or particular protein or it depends on the size, right? Like, you know, the nanoparticles, sometimes they can cross the blood-brain barrier. Once they cross its specific targets, the targets impacting some of your brain function proteins, it's going to cause damage to several diseases, including some of the Alzheimer's or Parkinson's also related to nanoparticles. So particularly when we looked at ultra fine particles, also pretty much like nanoparticles, we found out they are targeting specific pathways relevant to function of brain. Sometimes it's long-term exposure or chronic exposure or acute exposures matters to depends on what kind of a toxicity is going to induce. But in the long-term exposures, or sometimes that what do you call the different level of impact or mutation going to create, it's definitely going to affect the functions of various organs, including brain.
Okay, sir.
Yeah, it depends on which particle you are going to look for. Some of the particles are safe, some of the particle cannot cross the blood-brain barrier, and which can cause blood-brain barrier. And if you identify the particular targets, if it's going to affect that, you can really predict them.
Okay.
Yeah.
So,
Sneha, yeah.
Please, sir, go ahead.
Yeah, no. Sneha asked how one gets started with QSAR modeling. So, if you elaborate your problem, I can tell you an example. Like maybe I can tell you from what my example of QSAR study, right? The QSAR study is started in 1930 or 1972. In between, it's behaved like when we have an aromatic structure. That aromatic structure and how this aromatic structure, the properties are relevant. That's where people started using QSAR models. Like if you add some groups like hydroxyl group or various other groups, how the property is going to change, how the property going to change, how the stability going to change, how the stability that reflects activity is going to be toxicity. So if you want to develop some kind of a phenol based compound, like a benzene based compounds, right? Collect all the benzene and substitute benzene. That's where the Hanch and co-workers, they worked on and demonstrated these QSAR approaches. And predict the properties. That properties, what you are interested in, depends on what you're interested in. That time they are interested in some enthalpy or the enthalpy of evaporation or enthalpy or stability of the compound. That's where they started using substituted benzene. So collect list of structures, or right now using databases, you can also get some list of structures. And using that, you collect the experimental values if it's available. Create that's also relevant to your research. Then make them as a table. So now go to your computational approaches, like which methods are some of the trivial or non-trivial approaches, several of them there. Like if you go for quantum chemistry approaches, you take that molecule and minimize them into its bond length, prescribed bond length and bond angle of this particular structure. If you optimize them, then you will get that compute the properties like physical chemistry properties, like electronic properties, what we call them. The electronic properties are compared with the available experimental data. That will give you a relationship, right? Like how these computational predicted values and experimental values are matching with each other. That's where you create a model. That model can be validated using tests and training set. Once you have that more good, accurate, like linear regression relationship, or nowadays a lot of other machine learning, artificial intelligence tools are available to create the model. That model can be used for predicting the new set of chemicals which you are interested in. Okay, that's where you can start some QSAR modeling, or QSPR modeling, QSTR modeling, whatever it is, depends on what property you are interested in. Yes, sorry.
Yeah, so I guess there are no more questions. And if you have any query, you can, Sir has already given the email ID, so you can directly contact, or you can just drop down in WhatsApp group, or you can write it to me. I'll definitely direct to the sir. And sir, I'm really thankful to you. You took out time for this particular session, and we always look forward for your support for such future workshop.
Yeah, it's really delighted to be part of it, and thank you so much once again for giving this opportunity, and thank Anil Gupta ji and for his team, and also congratulations everyone, and all the very best to everyone for your greater success. Thank you so much.
Thank you so much, sir.
Yeah, thank you.